In this section, we will discuss the innate and adaptive immunity to viruses. As we know that our body responds to virus, both by innate immunity and adaptive immunity. Innate immunity responses against virus through interferons, natural killer cells, and antigen-presenting cells such as macrophage, wild T cell, B cell, and antibodies provide immunity against virus in case of adaptive immunity. Remember that innate or non-specific immunity is the defense system with which you were born. Adaptive or acquired immunity is a type of immunity that develops when a person's immune system responds to a foreign substance or microorganism. There is an interesting point arises here is that one of the antigens presenting cell called as dendritic cell. These dendritic cells form a bridge between the innate and adaptive immune systems. But a question arises that how dendritic cell forms a bridge between innate and acquired immunity? Well, you will get the answer of this interesting question later in this video. Let's discuss the innate immunity and then acquired immunity against viruses, or how our immune system works against viruses. When a virus infects a human cell, it takes over the cell's machinery and uses it to produce many more copies of the virus. Eventually, the newly made viruses burst out of the infected cell and go on to infect other cells in the neighborhood. It has already been discussed. Some of the weapons the innate system can use to defend against viruses while they're outside of cells. For example, proteins of the complement system can opsonize viruses for phagocytosis by macrophages, and neutrophils and complement proteins can destroy some viruses. However, once a virus has entered a cell to begin its reproductive cycle, these weapons are ineffective. These are cell if these cells have been infected by the virus then. The role of innate immunity starts through interferons. Role of interferon is that the interferons produced by these cells can actually bind to receptors on the cell. This binding can result in the production of several hundred antiviral proteins that act to reduce the amount of viruses produced by the infection. Although many cell types can produce type 1 interferons, the king of interferon, by far, is a white blood cell called as plasmacytoid dendritic cell. These interferons will be discussed later in detail. Natural killer cells has many roles. Let's discuss them briefly, and then they will be discussed later in this video. Natural killer cells can give off cytokines such as interferon gamma that help with the defense. In some cases, natural killer cells employ an injection system that uses perforin proteins to help deliver suicide enzymes, for example, granzyme, into a target cell. That will be explained in detail later with mechanism. In other situations, a protein called Fas ligand on the natural killer cell surface interacts with a protein called Fas on the surface of its target cell, signaling the target cell to self-destruction. Natural killer cell also produce kill and don't kill signals. That will be explained later in this video. Natural killer cell produce interferon gamma. They also produce perforin. They also produce granzyme. They have Fas ligand. It also produces kill and don't kill signals. Third is the antigen-presenting cells, such as macrophages and dendritic cells. Macrophages ingest free viruses through phagocytosis. Macrophage engulf the virus, and then lysosomes digest the virus and is removed from macrophage as a waste material. As macrophage is a antigen-presenting cell, so it presents antigen on its major histocompatibility complex. Other type of antigen-presenting cells are the dendritic cells. They also have major histocompatibility complex to present antigen on its surface. Dendritic cell is a bridge between the innate and adaptive immune system by presenting antigen to T lymphocytes. This was all about role of innate immunity. As we have already said that dendritic cell is the bridge between the innate and adaptive immune system by presenting antigen to T lymphocytes. If the innate immunity, which involves the interferons, natural killer cells, and macrophages, is unable to curb an infection, then the role of adaptive immunity starts here. As we have discussed that adaptive immunity involves T cell, P cell, and antibodies. Let's understand their role in adaptive immunity to viruses. Mature dendritic cells present peptide fragments of antigens to T cells through surface MHC molecules, which is signal 1, and also provide co-stimulatory signals through B7 family ligands, which is signal 2. Both signals are required for efficient T cell activation. Let's understand this. T cell interacts with antigen-presenting cells through T cell receptor, and CD4 on T cell, and MHC2 on antigen-presenting cell, which is dendritic cell. Other interaction is when CD80 and CD86 binds the CD28. Now, the pregulation of inducible co-stimulant occurs, and the B cell interacts with T cell through antigen receptor, presenting antigen to T cell through T cell receptor. CD80 and CD86 binded with the CD28. There is also interaction of inducible co-stimulant and inducible co-stimulant ligand, or also called as B7H or B7RP1. Now B cells produce the interleukin-6 that cause the generation of plasma cells that produce the antibodies. Now, role of antibodies starts here. So let's discuss the role of antibody. Antibodies can actually bind to a virus while it is still outside of a cell, and can keep the virus either from entering the cell or from reproducing once it has entered. Antibodies with these special properties are called neutralizing antibodies, and the process is called as neutralization. For example, some neutralizing antibodies can prevent a virus from docking on the surface of a cell by binding to the part of the virus that normally would plug into the cellular receptor. When this happens, the virus is hung out to dry, opsonized, and ready to be eaten by phagocyte. This is called as opsonization. Antibodies activate the complement system. This complement cascade is made up of series of proteins. It helps antibodies and phagocytic cells to clear pathogens. Together, the series of protein generate the membrane attack complex. C5b, C6, C7, C8, and C9 are membrane attack proteins. It causes infected cell to lyse and die. This role of antibody is called as complement activation. Another role of antibody is that antibodies bind to viral antigen on target cell surface. This is the viral antigen on cell surface, where antibody binds. So, this leads to activation of natural killer cells that has Fc receptors. These receptors recognize the bound antibodies, as a result of which cytokines are released by natural killer cells can kill infected cell. This is called as antibody-dependent cell-mediated cytotoxicity. That will be explained later in this video. This was all about the players of innate and adaptive immunity, and their role in providing immunity to virus. In our upcoming sections, we will talk about the roles of these players of immunity in detail. When we make an immune response to a given infectious agent, by definition, that microorganism must exist in our environment and we are likely to meet it again. It would make sense then for the immune mechanisms alerted by the first contact with antigen to leave behind some memory system that would enable the response to any subsequent exposure to that particular antigen to be faster and greater in magnitude. The higher response given by a primed lymphocyte population is due to the presence of memory T cell and memory B cell, which not only form a quantitatively expanded population of antigen-specific lymphocytes, but also are functionally enhanced in comparison to the original naive lymphocytes from which they were derived. This was all about the innate and adaptive immunity to viruses. In our next section, we will talk about interferons.